But before I get to that, I wanted to figure out what exactly was causing this difference. The difference between the two plasmids, PET28 and PET11, was essentially the selection marker, kanamycin versus ampicillin, or in our case, carbenicillin. And in one case, the beta-lactamase marker for ampicillin is secreted into the periplasmic space and overloads the periplasm so much that it gets secreted in the media. For the aminoglycoside phosphorotransferase, the kanamycin, that confers resistance to kanamycin, it stays within the cytosol and doesn't cause periplasmic stress. Initially, we had some tentative data that suggested that the TEM1 beta-lactamase, simply the presence of it in the periplasm, was causing some dysregulation that conferred sensitivity of the interior to exterior osmolytes. But when I did the experiment, simply subtracting carbenicillin from the final expression media, while we actually see an increase, at least a little bit, in the amount of coprotein produced, we completely recover the incorporation of the Z domains in the particles, which was kind of cool. I had completely neglected the fact that PMB1 origins are really good at sticking around, and we could do this. I'll touch that later. So when it comes to the sucrose, the mechanism that I think is in action, this is a really loose hypothesis, is one simply of molecular crowding. So here I've got mRNAs, those black lines, and these blue things are really crude ribosomes. And so if you think of the VLPs as assembling really fast off the ribosome, having really fast kinetics of assembly right off the ribosome, and the Z domain extension fusions not assembling, they don't, you can express them, they don't form VLPs. It becomes, increasingly apparent that diffusion is what controls the incorporation of Z domains into the particles. You have to produce enough Z domains saturate the cytosol such that they're present at the ribosomes where the regular coprotein is being produced so that they can be assembled into VLPs. When you add sucrose and you have carbenicillin around, which disrupts the periplasmic equilibrium, you expose the interior of the cell to greater osmotic pressure and you increase molecular crowding. So if the coprotein is sampling a bunch of conformational space and interaction motifs, increasing molecular crowding increases the contact time between the proteins and should theoretically increase the rate of assembly, which would explain why we see fewer Z domains per particle, because it simply doesn't have time to diffuse to the site of assembly. The sodium chloride result is a little more confounding, but it turns out that sodium chloride actually acts as a metabolic poison before it acts as an osmolytic poison. And so my hypothesis on that is that an increase in sodium in the cytosol, rather than potassium from simple osmotic stress with the sucrose, results in some sort of change in the association with the internal RNA of the capsid with the coprotein. And so what we see is perhaps a slowing of this because the sodium is interfering with the association between the RNA and the coprotein. So what conclusions have I drawn from just these few short slides? The E. coli cell envelope manages a pretty high periplasmic load without much stress. You can fill the thing up with TEM1 beta-lactamase and really nothing goes wrong. The E. coli cell envelope is dysregulated by carbenicillin or its hydrolysis product. Obviously I couldn't differentiate between the two in the experiments I performed, but the simple presence of the antibiotic is enough to screw up regulation of osmolytes. Dysregulation of the envelope exposes the cytosol of E. coli to osmotic stress effects, which we observe as an output in the number of Z domains in a particle. And finally, that the bacteriophage Q-beta fibrous-like particle assembly process is sensitive to these effects. So this is really interesting for us from an engineering sense because it means we can start fine-tuning the number of things that we expose on the outside of the particle. And this is really key when we want to start loading antibodies and stuff like that on the outside. So at this point, there's a lot of questions that I really, really want to ask, and I simply don't have time to. Is the beta-lactamase, for one, induced by carbenicillin or its hydrolysis product? Obviously, a simple normalized Western blot would probably answer this question, but it's kind of getting into some more discovery biology that gets away from the engineering that we're interested in. But it is a valid question. If beta-lactamase is upregulated by carbenicillin, it's still possible that TEM1 beta-lactamase is causing this dysregulation. Next, I've been wondering if the SOS response is responsible for part of this. The SOS response is known to shut down, at least temporarily, the E. coli cell division in response to beta-lactam antibiotics. And so I've been kind of curious to get an output on this to see if we're activating that, even despite the fact that we've got TM1 beta lactamase present. And the other thing, which is a big issue that I didn't realize until I started researching it, the beta lactamase permeability through the membrane is a really big deal. There are a lot of papers that exist studying the permeability of the membrane to beta lactams. And I'm starting to wonder if perhaps the beta lactam getting in actually screws up the membrane enough to let other solutes in and stuff like that. So there's a lot of interesting biology questions yet to ask, but the engineering implications are pretty obvious. So at this point, I'd like to thank a few people. First of all, a second year in our lab, Jason Fiddler, for corroborating my initial sucrose results, because initially I thought I was just a horrible experimentalist. Dr. Andrew Udit, who was a postdoc with us, actually did some extensions of these experiments with peptide inserts. Unfortunately, I couldn't present any of his data today. And Peter Smith in the Romsburg lab for what were probably nothing to him, but exceptionally helpful discussions for me. I'd like to thank my advisor, Professor M.G. Finn, for being very patient with me as I go off on these wild tangents, which are kind of sometimes unrelated to what we're actually interested in. And finally, collaborators with us on the Q-Beta project. And someone who tells me a lot about history and lets me learn from my mistakes, Professor Romsburg, Professor Schneemann for collaborating with us on different fusion expressions, and Professor Manchester for collaborations with CPMD that validated some of our ideas about the particles. The NIH for funding the work, and you for your attention. I'd be happy to answer any questions. I know it's close to lunch, it's cool.
What applications do you see for decorating
Oh, they're practically limitless. Already with the Z-domain, we've got some materials that we make. So if we mix high concentrations of the Z-domain particles with IgG antibodies, we get this sort of amorphous precipitate, which we're actually characterizing by NCM right now, and we're interested in that as sort of biodegradable, implantable delivery device for antibodies. In addition, there are some really interesting bivalent IgG papers out right now, or not IgG, but single-chain antibodies, that I think it was in Science, where they were able to completely eliminate a tumor with like 6 micrograms of bivalent CFB. And so we're kind of interested in exploring that possibility, like targeting immune cells to cancer tumors. In addition, work with Aneta's lab with CMG2 has implications for anthrax vaccines. Jason actually is working on large catalysts, displaying catalysts on the outside and using those in process chemistry. It just goes on and on and on.
The others? If not, please join me in thanking all speakers. We have lunch now. We're back at 1:45.
Yeah.
That's right.
Yeah.